Welcome to Dr. Balga's podcast for the Curious Doc. Today is May 2nd, 2026. This video is in a Nature Medicine article published online a few days ago on 21st April 2026 titled Epigenetic Fingerprints Link Early Onset Colon and Rectal Cancer to Pesticide Exposure. There's an accompanying News and Views article published with the title Uncovering Risk Factors in the Exposome for Early Onset Colorectal Cancer. Colorectal cancer is rising disproportionately in people younger than 50 years. Early onset colorectal cancer shows more rectal and left-sided tumors, more metastatic disease at diagnosis, and aggressive histology, while genomic alterations are broadly similar to older onset colorectal cancers. The hypothesis is lifestyle and environmental exposures across life, the exposome, leave DNA methylation fingerprints, and that may explain early onset colorectal cancer beyond classic risk factors. Study design: tumor DNA methylation study using weighted methylation risk factors as proxies for 29 exposome traits across lifestyle, air pollution, and pesticide domains. The discovery cohort comprised of the Cancer Genome Atlas colon adenocarcinoma, 31 individuals with early onset colorectal cancer below the age of 50 years versus 100 late onset colorectal cancers above the age of 70 years. Demographics: early onset colorectal cancer, 18 out of the 30 were female, that's 58%. Mean age 43 plus or minus 5, whereas late onset colorectal cancer, 43 out of 100, that is 43%, with the mean age 78 plus or minus 5. In the replication cohort, meta-analysis of 9 independent colon, rectal, and colorectal cancer methylation cohorts, 83 early onset colorectal cancer versus 272 late onset colorectal cancer. Arrays mostly Illumina, human methylation 450, and 1 EPIC cohort. Exposome features 11 lifestyle factors, 4 air pollution particles, and 14 pesticides. 16 CpG island marker sets were derived from epigenome-wide association studies or EVAS using genome-wide, and false discovery rate thresholds were used. Statistics: multivariable logistic regression comparing early onset versus late onset colorectal cancer, gender adjusted when feasible. Replication by cohort level meta-analysis. Additional robustness models adjusted tumor purity, tumor location, family history, race, and microsatellite instability status. Duration follow-up: no individual longitudinal follow-up. Population validation linked pesticide use intensity to early onset colorectal cancer incidence across 94 US counties over 21 years. What did they find? They found that methylation risk scores confirmed known early onset colorectal cancer associated patterns, including educational attainment, diet, and smoking, and identified herbicide picloram as the new candidate risk factor. The limitations of the study: observational and proxy based. Methylation risk scores mark exposure fingerprints, not proof of causality. Direct picloram methylation data in humans was unavailable. Pesticide EVA source may limit generalizability. Self-reported pesticide use and male farmers of European ancestry. Small early onset colorectal cancer sample sizes, and the data sets were heterogeneous. The key takeaway of the study: the study triangulates tumor epigenetics, gene expression, and population pesticide data to nominate picloram as a plausible testable exposomic contributor to early onset colorectal cancer. Clinical action today is vigilance and prevention, not pesticide panic. The News and Views article discusses the epigenetic signal, its skepticism, and the next steps. Clinical pearls: early onset colorectal cancer is not just colorectal cancer occurring early. It has distinctive clinical pathologic features and may reflect life course exposure windows. Methylation risk scores are molecular smoke alarms, useful for hypothesis generation but not yet diagnostic tests or causal proof. Picloram is the headline signal, not the whole fire. The broader message is that the exposome, diet, smoking, socioeconomic context, pollution, and pesticides may shape early onset colorectal cancer risk. The strongest clinical interpretation triangulation, discovery methylation signal, replication meta-analysis, gene expression correlation, robustness testing, and county level ecological validation all point in the same direction. For clinic visits, ask about family history, rectal bleeding, iron deficiency, changes in bowel habits, unexplained weight loss, smoking, diet, occupational and agricultural exposure, and residential pesticide proximity when clinically relevant. Do not change screening solely because of suspected picloram exposure. Follow current colorectal cancer guideline screening guidance, but lower the diagnostic threshold for symptomatic young adults. The research priorities are direct exposure measurement, normal tissue methylation studies, dose response biology, reversibility of signatures, diverse cohorts, and mechanistic models of colorectal carcinogenesis. Bottom line, for physicians, the exposome is becoming measurable. The next frontier is moving from fingerprints to fingerprints with fingerprints. Exposure, mechanism, and prevention aligned. This is an infographic of the article. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.